Cell phone PMF to break up Alzheimer's, amyloid. Come on, what are you talking about? And I got this image here of our little beta amyloid from the protein database. It's the code is 6WZO. Gosh and others were the author of this image, published in 2021. Okay, and this is, again, amyloid beta 1 through 40 fibril derived from Alzheimer's disease cortical tissue. And they seeded it some way or another that you can read about in this publication. And what we can see is that the aggregation nature in this particular model. And here is our amino acid code. And we can see a lot of hydrophobic amino acids kind of stacked together, like phenylalanine is one of them. So you see these dark colors here, kind of aggregating. So this is kind of how you form amyloids. And this slideshow is going to document the work from Gary Arendash's lab, showing counterintuitively that this cell phone EMF can break these things up. And the other thing of personal interest to me is these histidines here in purple. And histidine can also bind copper, and you may have heard of the association of copper with amyloid formation, so this will be part of our story. Electromagnetic field treatment protects against and reverses cognitive impairment in Alzheimer's disease mice. Now, what? Remember back in the early 2000s, there was studies show the weak association between cell phone usage and brain tumors and Alzheimer's disease. And here's the cell phone, the position of the hippocampus, this memory part of the brain that is affected by Alzheimer's disease, right close to the cell phone back in the days where smartphones were less popular. And this study showed long-term EMF exposure directly associated with cell phone usage. This is 918 megahertz at 0.25 watts per kilogram provides cognitive benefits to this Alzheimer's disease mouse model. And this enhancement was seen in the normal mice genetically engineered, as well as the mice genetically engineered to develop a human version of familial Alzheimer's disease. In Alzheimer's disease mice, long-term PMS exposure reduced brain amyloid beta deposition / aggregation. And this led to the formation of... Okay, let's take a look at one of the proteins that's mutated in this Arendash model of Alzheimer's. And the mice they used had a mutation in the amyloid precursor protein. And this is from Swiss Prot, and the mutation was right before this cleavage site by beta secretase. So here's the amyloid precursor protein, and this is beta secretase, an enzyme. And the Arendash group really concentrated on the mitochondria in the study I'm going to present in this slideshow. And this came from Wilkins, the interaction between the amyloid precursor protein and the mitochondria. Here are some highlights of this paper: that the amyloid precursor protein is in the mitochondria-associated membranes connecting calcium stores of the endoplasmic reticulum and the mitochondria. And under normal circumstances, which is very interesting, atom 10, also known as alpha secretase, cleaves at this site rather than beta secretase. So this particular mutation associated with Alzheimer's disease might make cleavage by beta secretase more attractive. And instead of getting the C99 product, with the normal cleavage, one gets the C83 product. And synaptogenous neurite extension promoting soluble APP alpha instead of the APP beta as we see here in this cartoon. This is the basic protocol of Gary Arendash's publication in 2012. And he started out with 15 to 17 months old APP Swiss 1 PS1 transgenic mice containing these mutations. A mutation of lysine 670 to asparagine, and a mutation of methionine 671 to leucine, leading to elevated levels of A beta 1 through 40 and A beta 1 through 42. And recall that these two regions, 670 and 671, are really close to the beta secretase cleavage site that leads to the A beta peptides. And they use a generator for 2 hours period of electromagnetic waves per day in the early morning and the late afternoon. And the EMF exposure was at 918 megahertz, a radio frequency with carrier burst repeated every 4.6 milliseconds, giving a pulse repetitive rate of 217 hertz. And the electric field strength varied between 17 and 35 volts per meter, and this resulted in a calculated SAR levels that vary between 0.25 and 1.05 watts per kilogram. And this state 2 respiration, and technically this is the basal rate of respiration, and it is measured as oxygen consumption when they add stage 2 respiration. And to avoid copyright infringement on these bar graphs, I did something fun. So I basically drew arrows on the top of the bar graphs and traced them back to the y-axis for oxygen consumption. In neurotypical mitochondria from the cortex, we have an oxygen consumption here. And add EMF from the cell phone frequency, it's here. In the Alzheimer's disease, cortical mitochondria, the oxygen consumption is a lot less. And cell phone EMF does not help much, but it's statistically significantly more. And here we are in the hippocampus, neurotypic plus EMF, we get more oxygen consumption. Alzheimer's disease, it's a lot less oxygen consumption in the mitochondria, and the cell phone EMF really does boost mitochondrial oxygen consumption. So let's try ATOM, neurotypical mice here with cell phone EMF a little bit more, but not significantly more. And then Alzheimer's disease and with cell phone EMF, not that much difference. And in the amygdala, neurotypic is here, plus cell phone EMF is here, and Alzheimer's disease and state 5 maximum respiration, which is measured by addition of 1 millimolar FCCP, which sits in the membrane and transports protons so that the proton-powered ATP synthase does not work. And here we have protons going through the membrane that disable a maximum state 5 respiration. I wanted to have a picture as to why we care. Well, we care because neurotransmission is hard work, and there are a lot of mitochondria in the presynaptic membrane. Okay, let's look at some data. In the cortex, neurotypic, we have a fairly high maximum respiratory rate. And cell phone EMF improves things just a little bit, significantly. Alzheimer's disease is a lot lower, and cell phone EMF does not bring it back to control levels, but it improves things a lot. and the hippocampus, mitochondria. Neurotypic is here, and cell phone EMF improves things slightly and significantly. And Alzheimer's disease mitochondria from the hippocampus have a much lower maximal respiratory rate. And add cell phone EMF and you get a lot more. And neurotypic, and add cell phone EMF, no change. Alzheimer's disease and the striatum. And cell phone EMF doesn't really improve things in this particular part of the brain. And in the amygdala, neurotypic, cell phone EMF, no change. Alzheimer's disease plus cell phone EMF, no change. And why it's brain region specific, I do not know. I don't think it was known at the time of this publication, but this same group has published a lot more that I hope to make videos of. Complex 4. Complex 4 is where complex 4 activity from mouse brain mitochondria. In the cortex, the oxygen consumption rate is here for the neurotypic. And if the mice had been treated with cell phone EMF, it causes a slight, significant increase in oxygen consumption. And in the Alzheimer's disease mice, the cortical complex 4 activity is a lot lower, and it is significantly improved by cell phone EMF treatment. Moving on to the hippocampus, neurotypics here, and plus EMF causes a slight increase in the oxygen consumption rate. And here we are with the Alzheimer's disease. It's a lot lower, and the cell phone EMF really boosts the oxygen consumption rate in complex 4 of the mitochondria. And in the striatum, neurotypics here, the cell phone EMF improves a little bit, and in Alzheimer's disease, it's a lot less, and again, the EMF from the cell phone does increase the oxygen consumption a little bit, but not as much as in the hippocampus. And in the amygdala, we have this for the neurotypical mice, and really the cell phone EMF does nothing to change the respiration. And Alzheimer's disease and EMF is really the same. And let's just remember that copper is part of complex 4, and it can also possibly bind the beta amyloid. Mitochondrial respiratory control ratio. This is the respiratory control ratio, RCR, was determined by dividing the rate of oxygen consumption for state 3, which is determined by addition of ATP, by state 4, which is determined by oligomycin, an ATP-ACE inhibitor. And what this does is it tells us that values less than 2 for RCR can indicate poor coupling, and it can also indicate a proton leak, also known as the uncoupler protein, UCP. And UCP basically takes protons from the mitochondrial respiratory control ratio. Recalled from the previous slide that this uncoupler protein tends to let protons bypass the ATP synthase that makes ATP, and in the process of letting these protons bypass the production of ATP, it tends to heat things up. But let's just look at what it does to the respiratory control ratio. And remember, anything below 2 is generally considered uncoupled. And in the cortex, things are pretty good with a ratio around 6 for the neurotypical with and without cell phone EMF. And in the Alzheimer's disease mitochondria, things aren't quite in the bad zone, but they're not good either. And the cell phone EMF does help things. And in the hippocampus, the control ratio is pretty good in the neurotypic, and with EMF, it doesn't change things. And in the Alzheimer's disease mice, mitochondria in the hippocampus, the cell phone EMF does improve things quite a bit. Okay, and moving on to the striatum, here's neurotypic with cell phone EMF, and here is Alzheimer's disease with cell phone EMF, which we see an improvement. And the amygdala, neurotypic and neurotypic plus EMF, and Alzheimer's disease with the EMF of the cell phone frequency. We see an improvement in the respiratory control ratio in the amygdala. And as I mentioned earlier, a flow of protons through this uncoupler protein tends to heat things up. Well, what happens to the brain temperature? And if we have midday EMF exposure, there's no change in brain temperature. And if we have morning EMF exposure, the brain heats up almost 1 degree Celsius. And I should add that our body temperatures and I guess our brain temperatures tend to be the lowest in the morning around sunrise. And mitochondrial reactive oxygen species. Traditionally, this is thought to come from complex 1 and complex 3. So basically, instead of the electrons going to complex 4, the electrons go to molecular oxygen to produce superoxide. However, there's this thing called Fenton chemistry. So we have our little amyloid bugger, and they can take something like electrons from a ascorbate and redox cycle them, going from copper 1 and copper 2 to produce superoxide, hydrogen peroxide, and hydroxyl ions. And this was adapted from this paper that you may find at this DOI. And here we have the measurement of reactive oxygen species with this fluorescent probe called DCF. And the more fluorescence, the more reactive oxygen species. In neurotypic mice, mitochondrial cortex is not that bad. And PMF might reduce it a little bit, but not significantly. And the Alzheimer's disease model, the reactive oxygen species are really bad. And they are remarkably reduced by cell phone EMF. Let's go to the hippocampus. And neurotypical, not much difference. And when you look at the Alzheimer's disease mice, mitochondria from the hippocampus, it's pretty bad. And the good news is the cell phone EMF can really reduce the reactive oxygen species from mitochondria from the hippocampus. And the striatum, neurotypical is here, EMF doesn't do anything. Alzheimer's disease, a lot of reactive oxygen species activity, and EMF reduces it a little bit, and in the amygdala, not that much difference. Neurotypic here, and EMF, no difference. Alzheimer's disease, mitochondria from this part of the brain, a lot of reactive oxygen species, and cell phone EMF does not do that much in this particular region. And he's saying, well, I really reduced the reactive oxygen species from the cortex, says our cell phone mascot. Okay, and mitochondrial membrane potential. This one has many interesting ways we can look at this. I have chosen the mitochondrial associated membranes, which exist between the mitochondria and the endoplasmic reticulum, also known as the ER. And the more outside positive, the more protons are being pushed out of the matrix, which is what our electron transport chain does. and we need these protons in the matrix to make ATP. Okay, so they use a fluorescent probe called JC1 that concentrates in the matrix. And monomers are at low membrane potential, which I have as green circles, and we have aggregates at high membrane potential, which I have illustrated with stacked red pentagons. And our little amyloid bugger says, hey, I live between the ER and the mitochondria and the mitochondrial associated membranes. So what is EMF doing in this region that might help the membrane potential? Well, let's take a look. Here in the cortex of neurotypic mice mitochondria, we have a relatively high JC1 fluorescence. And we find that cell phone EMF really doesn't do anything. And in Alzheimer's disease, the membrane potential is a bit lower. And that cell phone EMF significantly improves the membrane potential. The hippocampus, neurotypic, cell phone EMF, no big deal, nothing. Alzheimer's disease, a lot lower membrane potential, and cell phone EMF significantly improves the membrane potential. And the striatum, no deal in the neurotypic mice mitochondria from this brain region. And in the Alzheimer's disease mouse model, there is a improvement. And the amygdala, neurotypic, cell phone EMF, Alzheimer's disease, cell phone EMF. No big deal. And the final topic is mitochondrial ATP. And this was measured with an enzyme called luciferase. This is the same thing we find in the butt of a firefly. It lights up, and you measure how much it lights up as a measurement of how much ATP is there. And the importance of ATP should be self-explanatory. So let's look at some results. And luciferase luminescence, neurotypic from mitochondria from the cortex, cell phone EMF, no big deal. Alzheimer's disease is ATP is a lot lower, and the cell phone EMF does a little bit to improve the ATP, but not that much in my opinion. Neurotypic, hippocampus, cell phone EMF, no big deal. Alzheimer's disease, mitochondria, ATP, and a slight improvement with cell phone EMF, which is significant, but slight. And in the striatum, neurotypic, plus cell phone EMF, no difference. Alzheimer's disease, cell phone EMF, a slight significant improvement. And in the amygdala, neurotypic plus cell phone EMF, no difference. Alzheimer's disease, ATP, mitochondria, this part of the brain, lots slower. And then in this case, cell phone EMF didn't do anything to improve the ATP. In conclusion, I made this video because I wanted to understand the mitochondria biochemistry of this publication. NeuroEM1250531 is a follow-up study by the company NeuroEM based on the original publication in this video. The company has shown that carefully aimed radiofrequency EMF can send these amyloids into the blood via brain vascular clearance. And it is my hope that this video will renew interest in mitochondrial mechanisms as well.